All right. So I would like to warmly welcome everyone. This is the first international meeting dedicated to considering the risk of developing a viral life and what should be done. And it's a true privilege and honor to host this event at the Pasteur Institute. And this is a very historical place to do this, and it resonates very deeply with the work of Louis Pasteur himself. And so I'd like to just take a minute to remind you of this fantastic discovery. It's not probably the best known contribution of Pasteur to science, but I think it could be argued that it might be actually the more important one. And it actually came at a very young age for Louis Pasteur. He was just 25, just graduated, working at the ENS, École Normale Supérieure, here in Paris. And he was tasked to study some interesting properties of tartrate, which were basically crystals that people were getting from wine production that you can sometimes see on the corks of wine bottles. And people had realized that a solution of tartrates that you obtain from wine actually rotates polarized light. And this is a type of instrument that Pasteur would have used for that, where you would put the tartrate solution here in the tube. Here you have a mirror that actually polarizes the light. And then here you have the reader with a crystal that you can rotate and see if the plane of polarization had changed. And Pasteur, there was a mystery at the time that tartrate from wine was rotating light, but the paratartrate that was obtained chemically would not. Also, these two were considered to be isomers. And Pasteur had a keen eye to notice that when he was looking at crystals of paratartrate, that they were actually chiral. And he called that dissymmetry. The word chiral itself came only like 50 years later. And so he had also the understanding that life was basically then producing the wine, was producing just, the grapes were producing just one side of the tartrate, but also later that microbes that consume tartrate would also only consume one form of the tartrate. And so something like 40 years later, the Pasteur Institute, where you are all sitting today, was created and is part actually of a very large international network of Pasteur Institute around the globe, and I think this also sets the stage for a meeting that is all about international cooperation. Why are we here today? It's because of the work done by an amazing group of people who had the foresight to bring this topic to the front, write this article in Science and the technical reports that you can download by looking at this, taking a photo of this QR code. And so just to remind everyone, we'll have two key goals today: communicate the current scientific understanding of viral life feasibility, risk, and emerging governance conversations to a broad audience, and to convene the diverse stakeholders to determine the next steps. So on the more technical aspects, we'll have 20-minute lecture with 10-minute Q&As. There will be 2 lectures, a short break, and then 2 more lectures. After that, there will be a quick reception in the modules. People will guide you to them. It's hot today. There is water available. Please hydrate. And we'll have the Q&A sessions conducted in a specific way. We won't pass a microphone to the audience, but what you need to do is that you should have received a flyer with a QR code at the entrance. So please scan that QR code. It will take you to an application where you can write down the question you're going to have during the presentations. And so this is for people in the audience. People who are attending remotely, please use the Q&A function from the Zoom application. And as you might have noticed, there is a live French interpretation. And just a quick overview of the agenda today. And so with this, it's my great pleasure to introduce Filippa Lentzos. She's a professor in science and international security at King's College London, and amongst her various appointments, she chairs the WHO Technical Advisory Group on the responsible use of the life sciences and dual-use research. And Dr. Lentzos is an expert on global biolabs, responsible science, and BWC verification. And so it's an honor to have your expertise here today.
Thank you so much for the kind introduction, David, and for congratulations to you and the entire team who's behind this initiative. I have to say it's tremendously exciting to be here, to see so many of you here today, also to see so many faces that I know. It's an incredible honor to open the symposium, and I'm excited for the next couple of days of discussions that we've got ahead. So the legacy of Pasteur, as David outlined, is extraordinary. His breakthroughs in understanding the causes as well as the preventions of disease laid the foundations of hygiene, of public health, and much of modern medicine. And as the discipline of microbiology evolved, new breakthroughs occurred. These contributed to the greater understanding of the science and also to the betterment of humans and the planet's biodiversity, ecosystems, and environments. One of these breakthroughs happened a little over 50 years ago when molecular biologists developed the first tools of genetic engineering. With those tools came the ability to rewrite the genetic code and to possibly create, in test tubes, organisms that had never before existed. And while this possibility presented unprecedented opportunities, it also presented significant risks. At the time, commentators in the popular press pointed out that it seemed as though molecular biologists had reached the edge of an experimental precipice that may ultimately prove equal to that faced by nuclear physicists in the years prior to the atomic bomb. Rising to the weight of that occasion, biochemist and future Nobel Prize winner Paul Berg of Stanford University, together with nearly a dozen prominent molecular biologists, wrote an open letter warning of the creation of novel types of infectious DNA elements whose biological properties cannot be completely predicted in advance. Published simultaneously in the pages of 3 leading scientific journals, Science, Nature, and the Proceedings of the National Academy of Sciences, this Berg letter, as it became known, responsibly called for an immediate suspension of certain classes of genetic experiments because of serious concern that some of these artificial recombinant DNA molecules could prove biologically hazardous. As I suspect many of you will know, in order to consider the potential hazards they had identified in the letter in more depth, Berg and others went on to organize the Asilomar Conference on Recombinant DNA in California in 1975. About 140 professionals participated, including not just biologists, but also doctors, lawyers, and others, with the goal of drawing up voluntary guidelines to ensure the safety of this new technology. In Berg's own words, from an essay that he published in Nature, the meeting sought to set standards allowing geneticists to push research to its limits without endangering public health. Since then, responsible life science research is often now taken to include a much larger set of values than simply safeguarding public health and progressing science. So, for example, the WHO's Global Guidance Framework for the Responsible Use of the Life Sciences, which I was part of developing alongside a large group of international and interdisciplinary experts, very much builds on the Asilomar endeavor by naming scientific progress and safeguarding public health as key values. But more tailored to the contemporary context, the WHO guidance framework explicitly values principles such as the responsible stewardship of science, our security, our biodiversity, and our ecosystems and environments, as well as values such as inclusiveness and collaboration, and social and intergenerational justice. Responsible life science is also more complex because life science itself is far more complex. Half a century on from Asilomar, recombinant DNA technologies have matured tremendously, and significant new capacities have been added to the molecular biology toolbox. Today's molecular biologists, alongside an array of scientists from converging disciplines, can now read, write, and edit genomes easier than ever before. It has become ever simpler to identify the genes underlying desirable, as well as undesirable, traits and functions. Systems biology and synthetic biology have provided unprecedented capacity to engineer plants, animals, and microbes, including pathogens. The ever-increasing capabilities of the life sciences have been matched by a proliferation of laboratories and other infrastructure necessary to perform high-risk and high-throughput life science research. There is a greater constellation of funding sources available for research, and there's a broader array of actors and agendas outside the traditional academic community that is involved too, from amateurs and student groups to corporations, nation-states, and superpowered individuals, actors both from the global north and from the global south. Capacities, and arguably the stakes, are significantly higher today than they were in the 1970s. Responsible science is more important, but also more challenging than ever. There is another story we can tell about biology in the 50 years since 1975. In the early 1970s, countries around the world came together to negotiate the multilateral treaty prohibiting biological weapons. The treaty extended the prohibition on the use of biological weapons in war, which was and continues to be enshrined in the 1925 Geneva Protocol, and which our host France is the custodian of. The BWC extended the Geneva Protocol's prohibition on the use of biological weapons in warfare to also cover the development, production, and stockpiling of biological weapons. And the image you see on the screen is from the council chamber at the Palais des Nations in Geneva, where the Biological Weapons Convention was negotiated and agreed. As that convention came into force in 1975, there were high hopes that the treaty would put an end to the misuse of biology and the life sciences. Indeed, that is one of the reasons why security was given so little focus at the 1975 Asilomar Conference. Biosecurity concerns, it was thought, had already been dealt with. While there are no states that claim to have or to rely on biological weapons for their national security, and there has been no use of biological weapons on the battlefield since the treaty came into force exactly 50 years ago, concern about biological warfare programs has not gone away. Before the Biological Weapons Convention, there had been significant programs by the major World War II combatants, including in my own institutional home in the UK and in our host country, France. But there were also significant breaches of the treaty after it came into force. This included the largest and most sophisticated biological warfare program the world has ever seen in the Soviet Union, but also more modestly sized programs like the Iraqi program and the much smaller South African program. And it's not only states and state-sponsored groups that have been and remain of concern. International terrorist networks, criminal groups, religious extremists, lone wolf scientists, biohackers, and other non-state actors are also of concern. Allegations of military biological activities in breach of the Biological Weapons Convention continue today. While many of the allegations today are politically driven and part of larger geopolitical power plays, science also forms a significant part of increased concern about the potential use of biology and the life sciences to deliberately cause harm. The Strategic Defence Review, published by the British Ministry of Defence just last week, for example, emphasized that engineering biology, while creating the potential to enhance the capacity of the armed forces through advances in medicine, healthcare, and well-being, and possibilities for new energetic and explosive materials, also presents avenues for enormous harm in the shape of new pathogens and other weapons of mass destruction. Even more recently, 2 days ago, the Deputy Foreign Minister of Russia said that in recent years, several countries have been actively pursuing research focused on engineering artificial microorganisms with specific traits, enhancing the capabilities of existing bacteria, viruses, and fungi, and amplifying their inherent characteristics. He went on to say, today, synthetic biology methods enable not only the modification of existing genes and genomes, but also the creation of entirely new biological systems by altering nucleic acid sequences. Now, while, as I said, many claims about biological threats are politicized and part of international posturing, I think it's clear that it's becoming ever more pressing to address the security dimension of biology and the life sciences. A summit to commemorate the 50-year anniversary of the Asilomar Conference was convened earlier this year, on the exact same February dates as the 1975 meeting, and in the same conference grounds on the Monterey Peninsula in California. The summit brought together some 300 participants from around the world, including several of you in the room, as well as a few who had attended the 1975 meeting, and in general included scientists from multiple disciplines, as well as conservationists, bioethicists, lawyers, former government officials, national security experts, and journalists. We were there to discuss current and projected developments in biotechnology, with a particular emphasis on issues of safety and security, of risks and benefits, of governance and of public interest and the common good. In short, we were there to discuss what responsible science looks like today. The key overarching question from the original Asilomar conference was also very present. Are there some experiments that are so risky they simply should not be done? And similarly, are there some technologies that should remain out of reach? So 50 years on, are we any closer to developing red lines around genetic engineering? Should we be? Setting boundaries around genetic engineering is a complex endeavor. The risk assessment or harm-benefit judgments that we use are not straightforward. And they depend on the selection of factors that are taken into account in the assessment, as well as on the particular contexts and individuals doing the assessment. Yet even with all this complexity, general agreement did form at the 2025 Asilomar Summit around a particularly risky line of inquiry. And that line of inquiry was mirror life, which is, of course, what we're all here to discuss today and tomorrow. Mirror life consists of life forms composed entirely of mirror image biological molecules. Scientists today are increasingly able to synthesize such mirror image biomolecules. Indeed, recent advances have enabled the chemical synthesis of mirror image kilobase length nucleic acids and large functional proteins. The synthesis of such mirror image nucleic acids and proteins themselves are not the concern. They have scientific and potential therapeutic applications. Instead, concern focuses narrowly on mirror image nucleic acids and proteins that are aimed at creating life forms composed entirely of mirror image biological molecules. Such mirror image organisms, or just simply mirror organisms, cannot arise from existing life. The incremental steps through which evolution proceeds would be unable to invert the chirality or handedness of complex biomolecules, let alone all biomolecules in an organism simultaneously. It is also exceedingly unlikely that we will encounter mirror life that has arisen independently. But with scientific advances, a mirror organism might be created in a laboratory. Creating such an organism, even as simple as a bacterium, would be a far more complex feat of bioengineering than has ever been accomplished. But progress on key enabling technologies is underway. The creation of mirror organisms in the lab, and there are several plausible pathways to do so with bacteria, would constitute a radical departure from known life. The key concern is that mirror bacteria, or mirror life more generally, might evade the immune system and other biological defenses that we and the rest of the living world have evolved, presenting potentially unprecedented risks to humans, other animals, plants, and the environment, including potentially making large parts of the Earth uninhabitable. On the other side of the scale, mirror life appears to offer few foreseeable benefits, other than potentially satisfying scientific curiosity and personal ambitions, drivers that are well and good in normal circumstances, but not when the potential harms are of the scope and of the magnitude that we're speaking about here. So returning to the Asilomar Summit a few months ago, it was broadly confirmed by the meeting participants that, as it stands, creating mirror life presents a clear red line that should not be crossed. And the entreaty that was issued from the meeting, and that will shortly be publicly released, unequivocally states mirror life should not be created. The statement came with a caveat, however, and that caveat was a recognition of the contingent nature of harm-benefit assessments. The entreaty's prescription of mirror life was qualified by the signers with the clause, unless future research convincingly demonstrates that it would not pose severe risks. As the context around harms and benefits change, red lines might change too. The risks of mirror life are not a completely new discovery. In 1848, right here at the Institut Pasteur, Louis Pasteur first discovered molecular chirality and speculated about the possibility of mirror life. By 1992, scientists warned in a letter to Science that mirror organisms would have built-in immunity to attack from normal life, and urged researchers to consider these matters in detail before getting started. Similar warnings appeared in Wired magazine in 2010. The scientific community had been informally discussing the possibility of risks from mirror life for decades. But until the possibility of building mirror life came into view more recently, as John will talk more about, nobody had done a thorough risk assessment of mirror life. In December 2024, a group of 38 leading researchers working in Brazil, China, France, Germany, India, Japan, Singapore, the United Kingdom, and the United States published an analysis in Science of potentially severe risks of mirror life. The co-author list included 2 Nobel laureates and a further 16 members of national academies. Many of the authors are with us here today, either in the room or online, and you are about to hear from 3 of them. The science paper was accompanied by a 300-page technical report that carefully outlined the problem and provided the first in-depth assessment of the potential risks. The discussion at Asilomar and the resulting entreaty built on and greatly benefited from the findings of the paper and that technical report. The main message of the paper was not, however, to solely raise the alarm about this new risk. The aim was also to suggest a path forward. The authors of the paper estimated that capacities to create mirror life are likely to be at least a decade away and would require large investments and major technical advances. The authors emphasize, therefore, that there is a window of opportunity to consider and to preempt the risks before they are realized. To take advantage of this window of opportunity, the authors encouraged all relevant expert communities to critically engage with the analysis in the paper and in the technical report. They noted in particular that they welcomed arguments and evidence about mirror life that they had not yet considered. They called on the global community, including scientists, governments, ethicists, security experts, industry, and civil society groups, to collaboratively consider the risks from mirror life and to establish governance mechanisms that are capable of preventing the creation of mirror life, but that do not unnecessarily impede scientific research. To help facilitate greater understanding of the risks associated with mirror life and to further progress on governance, the authors announced a planned series of public dialogues on these topics in 2025. So today is a momentous day. It is the inaugural dialogue event, and you are all part of it. Please do make your voices heard. Our discussions will only be as rich as the contributions allow it to be. And the greater the range of perspectives, the better. Thank you all in advance for your participation. And on that note, I wanted to say how exceptionally pleasing it is that the Pasteur centers from around the world are able to join us online. It is good to have you with us. Finally, if you will permit me a personal reflection. I was not involved in the science paper, nor in the multi-month research effort that went into producing the accompanying technical report. But on the eve of their publication, I was invited to comment on them and on the wider initiative by the New York Times. I told the journalist that I was impressed by the paper, and I said, the article presents a careful consideration and thorough assessment of possible risks by a cross-disciplinary group of experts. They present their deliberations and conclusions in a factual way, in a top-tiered journal targeting the wider scientific community, but also inviting engagement from a broader range of stakeholders, including policymakers, research funders, and industry. They present a series of recommendations that are sensible and with which I agree. I share their view that the risks of creating mirror life cannot be justified by the relatively limited potential benefits, and that mirror bacteria and other mirror organisms should not be created. So to close, I want to come back to the WHO Global Guidance Framework for Responsible Use of the Life Sciences that I spoke of earlier, and whose associated technical advisory group I chair. The values and principles we developed for the framework speak of the need to responsibly steward science. We elaborate how this involves commitments to exercise caution, to identify, manage, mitigate reasonably foreseeable, potentially harmful consequences of basic research, and to develop and support policies that reflect the wider community's values, priorities, and risk-taking strategies, among many other commitments. The Mirror Life Initiative has so far embodied all of these aspects of scientific stewardship, and to my mind, the initiative constitutes an exemplary role model of responsible science today. And with that, I thank you for your attention.
Thank you so much, Filippa. Hi everyone, I'm James Smith. I'm adjunct faculty at the J. Craig Renter Institute and the Deputy Director of the Mirror Biology Dialogues Fund, which is helping to organize this meeting along with the Institut Pasteur. Thank you so much, Filippa, for that talk. Just a brief reminder, the way to ask questions is by scanning the QR code on the back of the agenda document that you should have all received when you came in. Some people have already been asking really good questions in there, but please do keep them coming. And I can come back to technical questions if you have them in future slides with different presenters. So Filippa, perhaps we could start. Could you maybe talk a bit more about the process through which you got to agreement on the WHO's global guidance framework on responsible use of the life sciences, and perhaps any lessons that we could take forward into this discussion about Mirror Life?
Thank you, James. The process actually mirrors the process that you've set up here quite nicely. It took 2, possibly even 3 years, and I believe some of the WHO team are also present in the room who did most of the legwork behind that. But it did involve first consultations with the stakeholder groups, so funders, publishers, institutions, and so on. It then involved outreach to individual experts, developing working groups of a number of working groups, and I know there are also people who were involved in those working groups present here in the room today. In my own working group, for example, we held several meetings, and I mean dozens of Zoom meetings to agree. It's hard work when you are trying to do something with an international audience, speaking across disciplines in very different contexts, to find areas of agreement. And it takes time. And so I think from that experience and all of the consultations that the WHO then took, and they took ownership of the final framework and guidance framework, but all of that experience, I think, is to a large extent baked into what you're trying to do here. So it's feasible, absolutely, but it requires a lot of hard work. It requires commitment, and it sure requires funding too. So I think some of those lessons that were and experiences that came through that process could also be well applied here.
Great. So we have a couple of related questions, which are along the lines of, there are many serious risks happening right now that we could be working on, people in this room could be working on. Why is this being discussed now? Why should it be a focus now when there are other priorities that we could be thinking about?
I think there are lots of priorities that we could be thinking about, and there are lots of risks that many of us are working on. I think in terms of Mirror Life, we see that there is this particular window of opportunity, and we see that the risks are particularly strong or heavy or weighty, if you like. And so I think there is a case to be made that here is a particular kind of, or a particular line of inquiry that we can all coalesce around and find agreement on, that in this particular instance, the risks seem overwhelming compared to the benefits at this point in time.
Great, thank you. So one more question. I think we've talked a lot about the risks here, but it's also, of course, very important to make sure that the benefits of scientific research can be protected. So how can we ensure that enabling technologies related research are not over-regulated in this case?
I'm sure that's a question that many of the scientists in the room are particularly interested in and have daily experience of fighting the red tape. I think the most important thing is first to recognize that what we're talking about is a particularly narrow piece of scientific investigation or line of inquiry, and to make sure that any rules or regulations are specific to that very narrow field of research. And equally, I think what's important is not just to make rules, create rules and regulations for the sake of red tape, right? You want to do it because there is general recognition that this is the right thing to do. You need to create that buy-in along the way, which is a negotiation process, and it involves giving and taking. and it's a long journey, but at the end of that, you have a product or a set of guidelines or an outcome that is more believed in and that can become part of the culture rather than yet another kind of tick boxing exercise that I know many of us struggle with.
Great. I think with that, we'll move to the next speaker. So,
Thank you.
Thank you, Filippa. All right. So the next speaker is Professor John Glass. John is a professor at the J. Craig Venter Institute and leader of the JCVI Synthetic Biology Group, the director of the La Jolla campus there. He led the research that created the first bacterial cell with a minimal synthetic genome. So John is a real leader in the synthetic cell research world. Together with Jack Shostak, he co-chaired the working group of authors that ultimately led to the 2024 Science article on Mirror Life and the accompanying technical report. So over to you, John.
Thank you, James. That pretty well covers the first couple of slides. Bon après-midi. As James said, I'm John, and I want to talk to you about how mirror life might be made. And I will note that the slides that I'm going to show are, in fact, the work of many scientists, and that collectively we all think this is a bad idea. So that said, we are not the first to think about the potential risk of mirror life and the possibilities. Although I think the technical report and document that we produced is probably the most in-depth analysis of this field. Previous eminent scientists who have spoken about this or written about this include Louis Pasteur, has been mentioned previously, Isaac Asimov from who has written about this, and of course my friend and collaborator George Church. Most interesting, I think, or also interesting, is a 2010 Wired paper where has the provocative title shown here, discussing the risk of mirror bacteria. In my own journey to talk about and think about mirror life, it first began when I learned that in college that life on Earth has a certain handedness or chirality. And this was rekindled when I became a synthetic biologist a couple of decades ago. It seemed an almost certainty. I should be on the right slide. Yes. It seemed an almost certainty that if we were to make an exact copy of, say, E. coli, but with mirror chirality, mirror proteins and nucleic acids, it would almost certainly work. Because I think I should also note that in 2019, I was part of a U.S. National Science Foundation ideas workshop that assembled about 80 synthetic biologists to come up with novel ideas that could advance the synthetic biology field. And one of the ideas we came up with was to produce mirror life. Not a single person in our group, in the room, had any objection when we thought this was a great idea, which I think shows something about how siloed biology and science is. Now, had infectious diseases scientists or immunologists been in the room, that result might have been different. In any case, I was very proud to have been part of the technical team that produced our that produced our science paper and technical document. Some of the authors of that are in the room, and this is, I think, an important achievement and may have influence in how we move forward with biology research in general, and certainly how we think about this topic. So, chiral molecules. What is handedness? Sorry, bear with me here. What is handedness? Think about your left and right hands. They are virtually identical, but a left glove won't go on a right hand and vice versa. So in life on Earth, proteins have a left-handedness based on how they refract light, and nucleic acids have D-riboses, which refract light in the opposite way. As a result, all living organisms almost exclusively have only one or two possible configuration of each kind, proteins and nucleic acids. In the lab, it is possible to chemically synthesize both DNA and protein molecules of the opposite handedness, but the interactions of these molecules are extremely precise. So chirality can make a huge difference. A famous example is the drug thalidomide. We know that, I'm going to take my jacket off because it's a little warm up here. We know that a molecule of one-handedness is an effective morning sickness drug, but molecules of the other-handedness were able to cause birth defects in newborns. So, just as it's possible to build mirror image molecules in the lab, it is theoretically possible to imagine an entire life form composed of molecules of the opposite chirality. The most simple life form would be bacteria, and that is the subject of what we're talking about today, is building mirror bacteria that would be identical to normal bacteria. Why are I and my colleagues excited about the idea of building life? Well, first, it would be an enormous technical accomplishment, a tour de force that might teach us in some ways about the origins of life. But these organisms capable of producing mirror biomolecules might be effective tools for producing for producing therapeutics. Because mirror bacteria interact differently with the natural world, the risk of making them are that immune systems wouldn't recognize them, and proteases and nucleases wouldn't digest them. Additionally, in the environment, they would be invulnerable to bacteriophage and wouldn't be digested by the numerous higher life forms that graze on bacteria. Deepa and David will talk more about these topics. But would it be possible to produce a mirror bacteria? And the answer is yes, absolutely, although it might take 10 to 30 years. The most likely pathway by which a mirror bacteria will be produced, we think, would be through the convergence of at least two parallel fields of ongoing research. First, decades-long efforts to produce more and more complex mirror biomolecules chemically for beneficial applications, and second, in my own field, where we're trying to produce synthetic cells by combining non-living parts to make a living cell. So what do we envision these two fields combining would allow? Is that it would allow the assembly of a mirror cell from non-living components. I'll talk more about this. Bear with me. What do we need to build a mirror cell from non-living parts? We need four things. We would need a mirror genome, or really, what do we need to produce any cell from non-living parts? We need a synthetic genome. We need to produce an effective central dogma system for protein production and RNA transcription, and we need a membrane to encapsulate both of these. And then additionally, we would need a method to boot these things up. Okay, while it is, the methods for producing synthetic genomes have been established, as well as the methods to produce effective membrane vesicles. What is not so straightforward is the idea of producing at least a mirror cytoplasm. We can do this now for natural chirality things, but a mirror cytoplasm is nowhere close to being done. And then the other thing that is important to note is that no one yet has produced a organism of natural chirality, even composed of non-living parts, much less a mirror organism. So who's the closest, one would say, for all of this? It would be my group. So what we were able to do in 2010 is boot up a chemically synthesized natural chirality genome by installing it in a living bacterial cell to produce what we call JCVI Syn 1.0, shown in this cover of Science. This combined the three technologies shown. We can likely make mirror genomes and mirror membranes, but the idea of putting together, but the production of a mirror cytoplasm is still far beyond our capabilities. So to produce a mirror genome, what did we do? Dan Gibson from the JCVI was able to take synthetic oligonucleotides and combine them using basically three enzymes: a correctase that repaired errors in or cut assembled oligonucleotides that were just put together and naturally assembled. It would cut oligonucleotides that had errors, a DNA ligase to close nicks between these things, and a polymerase to close nicks between these things. And as shown here, he was able to produce in 2009 a completely synthetic, by in vitro methods, 580 kb natural mycoplasma genome. So this is likely not a problem. It's also worth noting that Chu and Zhu showed in 2022 that they could use a chemically synthesized T7 RNA polymerase to produce 1 kb reverse chirality nucleic acids. So how do we build a synthetic cell from non-living parts? So the transplantation thing, the idea is you take a chemically synthesized genome that encodes a antibiotic resistance marker. You would take a cell that you would treat with polyethylene glycol to soften the membrane, and calcium to mask the charge of the nucleic acid in the cell. These would be combined by basically a chemical transformation like you do for E. coli now. And then when the cells are returned to a growth environment and treated with tetracycline, the previously diploid cells would divide into cells, and the tetracycline would result in the death of the cell that had the natural chirality, the native genome, to produce a synthetic cell. So this works now. We also know that you can produce chemically synthesized membranes from chemicals off the shelf, and this is a doable technology. So how would you boot this up to make a living organism from non-living parts? Well, the way we envision doing this is you would produce cytoplasm by taking a culture of, say, Mycoplasma capricolum cells. To isolate the cytoplasm, you would install a plasmid that would encode a number of transporter genes. You would encapsulate this using microfluidic technology to produce vesicles containing this transcription translation system. You would then combine these in a couple of approaches, either using transplantation or this method for combining membrane encapsulated vesicles and containing genomes, and another vesicle containing the cytoplasm. And this, in theory, should produce cells in a couple of days. We'll see. Now, it's more complicated to think about producing a cytoplasm. Here, so for 30 years, scientists have been producing mirror proteins and mirror nucleic acids. It's worth noting that mirror nucleic acids, if you load an existing DNA synthesizer with mirror phosphoramidase, it will very nicely produce mirror DNA. But for mirror proteins, it's more complicated. So this technology has been developing for 30 years. Phil Dawson at the Scripps Institute of Molecular Biology has improved this technology significantly, but it's still a complicated thing to produce. Bear with me. In any case, what you see here is a mirror peptide can be produced that looks like, I should remember this, but I don't, that looks like VEGA, that is an effective inhibitor of the chemical processes VEGA is involved in cells. So this is a theoretical application. There are a number of companies that are existing now whose ambition is to produce mirror peptides for therapeutic applications. One of the things that the mirror peptides or proteins would have as an advantage is the capacity to avoid metabolic degradation and immunological reactivity. And again, the progress in the synthesis of chemically synthesized large DNA proteins is increasingly, the process is we are moving more and more to being able to produce these more effectively. It's also worth noting that what hasn't been done is to make an effective mirror ribosome, or actually only recently have people reported the synthesis of normal chirality ribosomes, which are essential for making any kind of a cell-free transcription translation system. So this, in fact, I think is the most difficult task in the whole idea of making synthetic cells, is that ribosomes are really cannot be done easily now, and it may take years to get there. But the first synthetic natural ribosome was reported earlier this year. And so with all of these technologies, so the genome, membrane, and cytoplasm and boot up system would give you a natural chirality synthetic cell. To make a mirror synthetic cell, you have to put on the complexity of making chemically synthesized nucleic acids and proteins, and this is really hard, but it could be done in the next 10 to 30 years. Okay, we may be able to produce a mirror organism first that poses no risk, such as my cell, the mycoplasma, this mycoplasma with a synthetic genome, JCVI-SYN 3.0. It would exist, but it would be of no risk because it cannot make mirror nucleic acids, it can't make nucleic acids, it can't make proteins, it can't make cofactors. So these things would pose no risk at all. But once you make the first bacterium with a chemically synthesized mirror genome, a mirror bacterium, it would use existing methods that would allow us to produce, say, an Escherichia coli, which is capable of potential unprecedented risk. The idea of effective biocontainment of anything is a myth. And so all natural biocontainment methods are vulnerable to human error or deliberate misuse. Physical containment has well-documented failures. Think about the Moscow flu or possibly even SARS-CoV-2. So that even, and in the case of mirror bacteria, a single failure would be unacceptable given the unprecedented risk posed by these organisms. And biocontainment measures also could be reversed should such an organism be made, where in the case of a synthetic JCVI Syn 3.0 could be, which is safe, could be engineered to be incredibly dangerous. So where are we with this in general? By default, advances in several labs would have to be converged in the next 10 to 30 years to allow the creation of a mirrobacterium. Once created, mirrobacteria would be difficult to robustly contain, possibly impossible to robustly contain. Today, technical barriers remain high, but preventing an opportunity to address the risk before. But what we want to do is prevent an opportunity to arise when this could be an incredible danger, and establishing governance and a governance will be combined with political and scientific efforts. With that, I'll note that more of this can be seen in our technical documents, and I'll stop and take any questions you have here.
Great, thank you, John. Loads of questions coming in. Thank you all so much. So you talked about the need to stop this area of research, but you also talked about the fact that one of your goals is to create natural chirality synthetic cells. Isn't it the case that if we could make natural chirality synthetic cells, we would also be able to make synthetic mirror cells? And how do you think about that?
Making natural chirality synthetic cells from non-living parts would be a critical enabling technology. Are we willing to go there? There are advantages. I don't see a future for biotechnology that doesn't include synthetic cells. By making synthetic cells, we could obtain enormous new capabilities to produce medical advances, materials that are impossible with existing life. But this is still an enabling technology. Where I see is maybe the bottleneck, the way we could stop this, is either in the production of fully synthetic genomes, because one would have to order a couple of 100,000 sextamers of reverse chirality to make a synthetic genome. That might be stoppable. Or possibly we should never make a synthetic mirror ribosome.
All right, thank you. Someone else asks, do many companies use mirror research? So I think this is asking, what is the kind of industry landscape here? And in particular, are there benefits of mirror life that we should be thinking about that I'm not sure were covered in the slides?
So chemically synthesized mirror proteins or nucleic acids may have therapeutic uses. I know there are at least 2 US companies that this is their ambition, and I cannot think of their names offhand. There are a number of research groups, academic research, pursuing the same goals. The same is true in other companies. In themselves, they pose no risk. Think about the sometimes mantra of synthetic biologists. Yogurt can make more yogurt, but shampoo can't make more shampoo. So chemically synthesized proteins or nucleic acids are equivalent to shampoo. Mirror bacteria would be equivalent to yogurt. Does that get to the question? I attribute the yogurt shampoo thing to Pam Silver. It's worth noting.
Great, okay. So one of your slides, you talked about the fact that you didn't think containment of mirror life was feasible. I think some people might disagree with that and argue that there are, in fact, biocontainment measures that could be implemented for mirror life that would make them secure. And someone has asked, that would make them safe, sorry, and someone's asked a question along those lines, whether there are technical approaches to make mirror life research inherently self-limiting, so that the research can still advance, and we can advance scientific understanding of mirror life, but avoid the potential for weaponization and replication.
Okay, biosafety level 4 in theory works, but it is subject to human error or it is subject to malfeasance. If we were to do this work in facilities that were built on Mars, where no materials that were in those were ever allowed to come back, nothing that went in ever came out, I would be all for that. I think this is still a really interesting project. But the idea of doing this, I think the risks are so great. Again, possibility, the risk is this factor of what are the potential consequences, what are the potential good things that could come from it, or what, and so deciding how big a risk this is. I can see a future for mirror molecules as therapeutics, but not for those made from living forms, because once you make the first living mirror organism, it's not that hard to imagine making one that could be this incredibly dangerous organism that could have adverse consequences for all higher life on Earth. Bacteria will be fine, but we eukaryotes that rely on our immune systems, we could be screwed.
Maybe it would be worth saying a few comments, John, about what the benefits of mirror molecules specifically might be. Why are they potentially interesting from a therapeutic perspective?
So in my pocket, I have an insulin pump, and I have to take insulin all day long. Most of you who are not diabetics, you have pancreases that have beta cells that produce insulin all day long. A mirror molecule that had the exact shape and functionality of insulin, I could take that once a year, twice a year, and it would contain my blood glucose levels the whole time. Now, granted, I have to get more insulin when I eat and stuff, but there are a lot of proteins that have therapeutic uses that we have to take every so often. With mirror molecules, they could be extremely long-lasting, and they would likely have no adverse immunological consequences. So this could open up a whole new area of protein therapeutics, or even nucleic acid therapeutics, that would be long-lived, not immunological problems, and could solve a number of diseases better than we can do today.
Great. I think time for just a couple more. So in your slides, you mostly talked about what I think would be called typically the bottom-up pathway to creating mirror life. And in your comments that you just made about synthetic cells and mirror ribosomes and possible areas for that would represent bottlenecks, would those bottlenecks apply to other pathways to creating mirror life? So particularly to the top-down pathway? And when, if at all, do you think the top-down pathway will become feasible?
What James is referring to as a top-down pathway, imagine we engineer existing ribosomes to produce proteins that contain both D amino acids and L amino acids. And so the genetic code, in theory, would allow this, although we would have to make 19 new for the D amino acids. We'd have to make 19 new aminoacyl tRNA synthetases. So that would solve the idea of producing proteins of reverse chirality using existing organisms. So that would be one step. But then to further make a top-down mirror life, then you'd have to figure out how to have initially both mirror nucleic acids and normal chirality amino acids. This might be doable. It might have some real great uses, but it would be really hard. And I think it's much easier to think about building mirror life by the bottom-up approach that I described.
Great. All right, I think last question, and just to say up front that all the questions that people have asked are queued up here, and some of them that I won't be able to get to in this session, I'll be able to return to after some of the other speakers' points. There are a couple of questions related to whether other technologies like artificial intelligence, quantum, things like this, could accelerate the timelines that you've laid out here. So for example, could AI biodesign accelerate progress here? Are there other AI tools that might accelerate the timelines that you laid out in this presentation?
I think all of this is imaginable. Not necessarily obvious, though, in the sense that we know what a natural chirality ribosome looks like. Now, we haven't figured out an efficient way to produce them now from synthetic parts. And could AI help this? Possibly, but I'm not really clear how. In terms of designing mirror therapeutics that could be really useful, so we've already seen what AlphaFold 3 can do in terms of designing proteins. Now, if I were to make an insulin with the same sequence of amino acids, of D amino acids that my L amino acid insulin in my pump has, that would have no use unless you made a mirror John. But could you imagine a D amino acid, a mirror protein with the exact structure or the enzymatic function of insulin? AI approaches might do that for insulin, for a vast array of proteins that could have therapeutic uses.
Great. Thanks so much, John.
Sure. Thank you, James. Thank you all.
So there'll be a short 20-minute break now. Please stretch your legs, get up, have a chat, and be back here at 4:30 for the next talk. Thanks a lot. All right, on to the next bit. So we've got two speakers this afternoon. The first is Deepa Agashe. Deepa is an associate professor at the National Center for Biological Sciences in Bengaluru, India. There she leads a research group working on adaptive evolution in bacteria and in insects. She's received a Women Research Excellence Award from the Scientific and Engineering Research Board of India and a Faculty Excellence Award for the Society of Molecular Biology and Evolution. She's also an elected fellow of the Indian Academy of Science, and she was a fellow co-author on the Mirror Life Science paper and an author on the technical report. Over to you.
Thank you very much. It's a pleasure to be here. I'm looking forward to learning a lot of things from discussions tomorrow and from all the talks that are yet to come today. David is speaking right after me. So, as James said, I'm going to tell you a bit about mirror-bacteria and how or why we think they pose a risk if they are released in the environment. So I'd like to start off by thanking all the co-authors on the report that hopefully many of you have already seen, particularly the chapter 8, which deals with environmental risk assessment and survival and how mirror-bacteria might spread if they were to be released. So many of these co-authors are here. It's really nice to meet them in person, some of them for the first time. I also want to thank the reviewers of the report because they really helped us get a few different ideas very clear in the report, and the rest of the co-authors on the science article, which again, many of you have seen already. So let me dive into what is it that I'm going to talk about. I'll first discuss just a few basic core principles of ecological spread that should apply to any species or population that you can think about. I will discuss in particular how mirror-bacteria might access nutrients in the environment and how or will they die. I will then discuss what effects these processes might have on ecosystems in the environment, and finally, what potential countermeasures we may have access to and whether these will be efficient or not. So just to begin with some very, very basic ecology that I'm sure you've learned in high school at some point. Population growth rates are effectively just the net outcome of birth and death rates, right? So for a population to be a stable population, no matter what size it's at to begin with, it has to have a birth rate that is balanced by a death rate. Then you're going to have a steady population size for quite some time. You can have declining populations where the death rate is higher than the birth rate, so more individuals are dying per unit time than are being born. And the one of interest to us in context of mirror life particularly is whether a population of mirror-bacteria can grow outside. For this, it would require a higher birth rate compared to its death rate. And this is what I'm going to address in the next few slides. So what are the factors that are relevant for birth rate? What decides what the birth rate of a population is going to be? Again, the quality and the quantity of food, as is obvious, as well as how much competition there is for those resources. It could be nutrients, it could be space, either of them. What about death rate? What's important for death is things like predation or infection. So what can eat you, whether it's eating you from outside or inside. And abiotic stresses, such as UV light, extreme temperatures, pH, and so on, right? In today's talk, I'm going to focus on food and predation and infection, that is interactions with some of the abiotic factors as well as biotic factors, because as we thought about this, our group figured out that these two things are probably much more relevant and important to talk about in context of mirrobacteria. So here are examples of what kinds of nutrients would be actually needed. And it's hard to generalize because there are bacteria of all sorts that are around right now outside, and native bacteria. And depending on what is the model for mirrobacteria that are used, if they're generated, the nutrition requirements would vary a lot. So we can imagine on the one end of the spectrum, photoautotrophs. As the name suggests, these are organisms like cyanobacteria, which can use light as their energy source, and carbon dioxide, similar to plants, everybody's familiar with. These are, in some sense, the easiest thing to imagine in terms of a mirrobacterium surviving outside, because these organisms require very little external nutrition to be able to grow, because they just need plenty of sun, plenty of CO2. It's there in, in the oceans. And so this is sort of very easy to imagine. If one is able to make a cyanobacteria-like bacterium, which is mirrobacterium, it should be able to survive in the oceans. The other extreme is the picky eaters, the chemoheterotrophs, right? These are organisms which will require organic compounds to both get energy and to get their carbon. And so we're going to discuss more these kinds of organisms for the rest of this talk, because this is much harder to imagine, right? You need to provide a lot more to these guys, even in the laboratory. So an example of this is E. coli, which many of you have heard of as a laboratory workhorse. And a lot of what synthetic biology is doing is using E. coli because just the amount of information we have is so vast. So what are the actual things that these guys could be eating if they are released outside? What would be certainly limiting is chiral molecules, which would be the wrong chirality because they're generated by existing microbes which are of a left or right-handed nature. So for example, amino acids would be left-handed, which are abundantly available. Sugars, many sugars like D-glucose, abundant, but not really accessible to an obvious, easy mirrobacterium. On the other hand, it's much easier to access achiral nutrients, that is, nutrients that don't have a handedness to begin with. These could be easily uptaken by even an E. coli-like mirrobacterium. Another option is that if somebody wants to engineer your E. coli mirror E. coli to be able to produce enzymes to convert between left and right-hand forms of a nutrient, then that bacterium should be able to survive much better outside. Let me give you some examples of these kinds of achiral nutrients and what concentrations or availability do they have in the outside world. So it's a long list. I don't want you to read all of it. But what's important is to notice that there are several different entry points in the normal metabolic cycles that microbes have, including fatty acids, alcohols, sugars, and so on. And this is a fairly long list, right? So any of these, how do we get this list, by the way, right? This is experiments done with E. coli in the lab, where people provide minimal media, a little bit of mineral, a little bit of everything else that's needed, but importantly, just one source of carbon or one source of nitrogen, right? And the question they've asked is, well, can E. coli survive with just that one carbon source, just that one nitrogen source? And the answer is yes. So that's why these guys have made it to this list. So, okay, fine, there's a lot of potential sources of nitrogen and carbon that are available in the environment, but what concentrations are they available in? So again, we scoured the literature to say, okay, what are these concentration ranges looking like? As you can imagine, it's a hard question to answer because there are many kinds of natural waters and many kinds of soils, depending on where you're drawing your soil sample from, etc. But we have some ranges just to help us think about this. And for natural waters, for example, for several of these nutrients where we have data, the range is about 1 to 1,000 micrograms per liter. And in different kinds of soils, it's 0.8 to 8 milligrams per liter. What does a typical E. coli need as its minimal requirement for growth in the lab? On the order of hundreds of microgram per ml. Of course, the numbers vary depending on what nutrient, what other conditions, and so on. But the important point to note here is that these concentrations that E. coli or its mirror form would need are not astronomically large. They're not so impossible that they will not be found outside or accessible. So together, all of this made us think that birth rates are not difficult to imagine being maintained at a positive number, right? So birth rates are not going to be zero for a mirrobacterium, even if it's like E. coli. Like I said before, if it's like Synechococcus or Prochlorococcus, which are photoautotrophs that are already dominant in oceans outside, much easier to think about. What about death rates? So the primary source of death in native bacteria outside in environments is predation from viruses, so the bacteriophages that infect bacteria. Protists like amoeba, which will go and engulf these bacteria and eat them up, as well as other bacteria that are also predatory, like Myxococcus, which is found in soils very commonly. And the way that all of these kinds of predation are going to work is there's going to be steps, as shown in the diagram here, involving recognition, tracking the prey, engulfing it, right, and killing it, digesting it, and then assimilating the nutrients. And the problem, as you've already sort of been alluded to earlier, the problem with mirrobacteria is that the chiral molecules that are involved in these recognition systems are not going to work for mirrobacteria because the chirality is opposite. So reversing the chirality in mirrobacteria is going to almost certainly cause complete immunity from phage infections. So no phages are going to be able to attach and recognize these as potential hosts, and so there's no killing from phages, at least in the early stages. These should also be resistant to uptake and digestion for these same reasons. Even if they are digested, they're kind of useless as a source of nutrients because those nutrients are going to be the wrong chirality, right? And so they cannot be assimilated to make energy or biomass by the predator who's eating these bacteria. So for all of these reasons, we think that mirrobacteria would have a very low death rate. What about chemical means of killing, right? So all of this was where there's physical contact between the prey and the predator, or the host and the parasite. What about bacterial warfare, which is fought, wirelessly, so to speak? So this warfare happens through chemicals, which are released in the environment, and then which goes and attacks the the prey bacteria. And these include things like antibiotics, which everybody's familiar with, bacteriocins, and other bacterial warfare systems. And so we went and asked, okay, how many of these depend on chiral molecules, right, to work? And you see that many of them do, which is why they're unlikely to work against mirrobacteria for the large part. There's only a few examples here in black that you see, or gray maybe, which have a chance of working against mirrobacteria. So overall, I hope you're seeing that very low death rates are going to result for mirrobacteria. And this should allow them to colonize suitable habitats, even though they have low birth rates. So this is just an example of what I showed you before. If there's a growing population, higher birth rates compared to death rates, for mirrobacteria, it would still be the same, but the depression in the death rate is going to more than compensate for the elevation, the suppression of the birth rate as well. What happens if these guys do get outside, right? We turn to invasive species of plants and animals, which all of us are already familiar with. They often lack predators as well, so there's no top-down control, and as a result have had really devastating impacts on many kinds of ecosystems. I give you just two examples here. One is lantana, which is a pretty ornamental that you must have seen in gardens everywhere around the world. It's from South and Central America and has made its way across all kinds of tropical regions in the world and decimated forests, including in India, where I'm familiar with this. So basically it takes over, and part of the reason it's able to take over a forest is because nothing eats it, right? So the herbivores have not evolved with this plant, and so they don't see it as food. If they see it as food, the toxins in there kill it. So it's not very good. Similarly, you may be familiar with the example of cane toads in Australia. These are big, juicy toads. And so they were introduced as a measure of pest control, right, in Australia, as well as other parts of the world. And the problem is, again, because they look very edible, reptiles and other predators will try to eat them, but they are poisonous. So they again kill the predators. They've almost certainly led to extinction of several other species of native taxa in many, many places. So we know that invasives are a really big problem, and we suspect that mirrobacteria are basically going to be behaving like invasives in many ways ecologically. Even if they can't colonize and grow amazingly, even if they just persist in an environment, we also think that's sufficient for them to move out into better environments, right? So they can be a reservoir from which they can spread more through animals, wind, and water. Human transport systems, as you know, humans are everywhere on this planet, and so our ships, our people, our animals, they're moving around a lot, and it's easy for bacteria to move with them. As a result, they can spread across terrestrial and aquatic ecosystems easily. And an important point to make here is that there can be repeated introductions of these bacteria in the same environments, even if they're not thriving there, and that could help maintain some steady populations for quite some time. One other thing that really bothers me as an evolutionary biologist is that these things will evolve over time. It's inescapable for life to evolve. And the problem is that mirrobacteria could then evolve to overcome some of the limitations that I mentioned before, right? If there are some nutrients they cannot consume at the moment, over time, given some population size from which they can start off, they can evolve to consume different kinds of nutrients, to evolve better transport systems, to evolve more efficient metabolism, or to resist abiotic stressors like extreme temperatures or pH, which would normally kill many kinds of bacteria. It's also plausible that other organisms can evolve in response to mirrobacteria, which then has all these other downstream effects where the sort of natural course of evolution and ecological processes changes. So I want to now move to ecological systems and ecosystem level effects that we might be able to predict for mirrobacteria. We focused on two of these examples in the technical report that I mentioned before. One is the loss of keystone species. So keystone species are these species in a particular ecosystem that have an outsized effect on the functioning of the ecosystem. So in this cartoon of a marine environment, a food chain in the marine environment, imagine we knock out the top predator, right? The next level is going to flourish and do much better. The next one after is not going to do very well because it's got somebody eating it a lot, et cetera, et cetera. So you can imagine these cascading chain of events that is going to really change how that ecosystem functions at different levels. As a result of these kinds of processes really being perturbed, we expect that primary habitats could also change. So imagine in a forest where a really dominant tree species is being affected directly by mirrobacteria, then if that tree is not doing as well, the forest ecosystem changes. Finally, we expect that geochemical cycling as a result of these effects could also be part of these cascade of events. A second way, or a second example of this, clicker does not seem to be working. Next slide, please. There we go, is thinking about soil microbiota, right? So imagine that mirrobacteria and soil environments, there are incredibly complicated and complex microbial soil communities that exist, and these are very important for maintaining nutrient balance and for nutrient cycling across the global ecosystem. So if microbes that are mirrobacteria go and compete or kill these native bacteria, we can expect to see really large changes in these kinds of nutrient cycles. And in turn, we think that these could lead to big agricultural or ecological losses. Next, please. So can we control this, right? Is there any hope at all? Can we ecologically manage these bacteria if they get out? Next, please. Okay, so again, we go back to invasive species to learn some lessons. We know that invasive species are incredibly difficult to control. It takes a while to find invasive species, and then there are limitations on how much you can remove them. Here's a plot just showing how over time, as an invasive species establishes and spreads in a particular location, the losses, the control costs really exponentially go up, right? So the hope is to find an invasive species and control it towards the left side of what you see in this plot, where it's less costly and less problematic to eradicate it. And as you can imagine, for a microbe, this is going to be a huge problem. How do you find something that is microscopic and is out in the water or in soil? And I want to just drive home this point with one example. So you know of E. coli or Shigella outbreaks that happen, right, regularly if you eat any salad. This is a worry. So how much does it cost to monitor for these kinds of known pathogens where we know what we're looking for? About $600,000 a day per company that provides these kinds of services. That's the estimate, right? So imagine the cost for something that we don't know how to find it. How do we kill it? And the novelty of Mirabacteria, therefore, is going to be a really big lead to a compounding of these problems that I just highlighted, with invasives just being an example. What about chemical means of killing these things, right? Could we spray something which would kill them? Yes, potentially. So there are enantiomeric antibiotics, which would have the right chirality. We could make them. You could deploy them. You could also send out miraphages, which have been engineered to be able to recognize the opposite chirality of Mirabacteria. You can imagine engineering plants or animals with genome editing to be able to recognize and respond or be defended against Mirabacteria. But the problem always, looking at our planet as a whole, is the scale of production that will be required, the distribution that will be required, and finally the evolution of resistance, which again is going to be inescapable. So to conclude, I think I've shown you today that we think Mirabacteria could colonize and spread through multiple ecosystems relatively easily. Animals and plants can be continuously exposed to these Mirabacteria as a result. And we think that whatever evidence we've seen from other invasive species, what we understand about bacteria in ecological settings outside, means that the effects on terrestrial and aquatic systems could really be quite damaging. And finally, that countermeasures are likely to be quite limited in their efficacy because of the constraints that we face dealing with a microbe which we cannot see. I'm happy to end there and happy to take questions. Thank you.
Great, thank you, Deepa. There are a few related questions in the chat around nutrient availability. So my understanding is that in many different environments, nutrient availability is very poorly characterized. And so how can we be sure that there are going to be sufficient nutrients for example, a Mira E. coli to grow in many different environments? And a related question is, wouldn't there be a fitness cost to having Mira chirality resulting in lower growth rates in these different environments, which I think you touched on, but perhaps you could expand on?
Mm-hmm. Yeah, so these are good questions and something that we thought about a lot while trying to put together this report. So first one is, how can we be sure that there's enough nutrient available outside for these things to grow? And the answer is we're not. We're not sure about it, right? The best thing we can do is to ask if it is plausible that there could be positive growth rates. And that's what we're trying to do here, because as the person who asked the question correctly pointed out, we don't have good numbers for every environment conceivable outside. We're barely scratching the surface in the last few decades of asking what are actual bacterial growth rates and death rates looking like in natural environments. We don't have those numbers. We don't know what nutrients are there everywhere, what nutrients are available to be used. So what we've done is then fallen back on what is plausible, and what seems plausible is growth. The second related question is there going to be a fitness cost, and then is that not a problem for maintaining positive growth rates? And again, the answer is yes, it is a problem. So we don't expect Mirabacteria to be able to grow as fast as native bacteria in most environments. That is almost certainly going to be the case as far as we can tell. But again, it's also just as clear that phages and predators are not going to be able to attack Mirabacteria, which means they also get a huge boost in terms of how little they die, right? Abiotic stressors, they are going to be equivalent with the native bacteria that exists, so that's not going to differentiate between native and Mirabacteria. And the major thing that's going to differentiate these two things or distinguish them is how fast and how much they die. And there, Mirabacteria, we expect, should have a huge advantage.
Great. In your concluding slide, you mentioned countermeasures. What about the potential to make things like miraphage that presumably would be able to infect Mirabacteria? Could those be used to control Mirabacteria populations? Are there other countermeasures that would feasibly work?
Yeah, so the thing is that all of these countermeasures would work in one localized area, right? You can imagine spraying one patch of soil with these miraphages, for example, right? Or with mira antibiotics. How are we going to do it across the world? That is really the problem. And because of the existing dispersal and transport-related exchanges that happen across the world, right? It's impossible that by eradicating my little piece of land here of Mirabacteria, I can guarantee they will not come back. They will come back. It's very easy for bacteria to disperse. So I think that's the really important challenge that we need to think about much more.
And so, just following up on this, if a miraphage was released as a method of biocontrol, could that not eradicate mirabacteria populations, or would it only be able to reduce them?
Right, so it again also depends on what populations we're talking about, what population sizes. So in a typical system where coevolutionary dynamics will get set up, that means that the phage and the bacteria begin to evolve in response to each other over time. It's conceivable that as the phage get better and better at killing the mirabacteria in turn evolve to get better and better at defending themselves from the phage, right? And so is that going to happen? Is that going to allow for eradication still? Hard to predict, but what we know from coevolutionary studies, that other systems where we studied this, is that it's not an arms race that is going to usually lead to extinction of any one species necessarily, right? So we don't know.
Great. So someone in the room is convinced by the risks, they're clear, but asks, well, what about benefits in the environment? Are there any benefits of mirabacteria if they were released environmentally?
I have to say we didn't really consider this very much. And I would have a hard time imagining this also, simply because given that we expect them to be kind of inert and not interact directly with anything else, I don't see what benefit we would get from releasing a new bug. I don't know, yeah. I mean, might be a fun thought experiment, but I would be very worried about doing it.
All right, let's just have one more. Maybe a more general question, asked from someone via Zoom. How can we prevent communicating the risks about mirror life from undermining public trust in engineering biology, and I think more broadly in science, especially at a time like this?
I would say that we're taking perhaps the best step possible in opening this up for discussion right now, and so I hope that this helps to build trust that scientists should and can be responsive and responsible to public concerns. In some sense, technology is already there, just like AI is already there. It's a bit late to think about, who's in charge, what are we going to do exactly, and how are we going to control it. It's here, we're already using it. Millions of lives are saved every year because of technology, biotechnology that's deployed to make medicines, to make vaccines, and so on and so forth. So how to use it responsibly is something that we all need to take charge of and discuss and think about, and hopefully we find some solutions. So I hope that public trust grows, not diminishes, in the value of science and in discussing scientific progress.
All right, thank you very much.
Thank you.
Great. So our next speaker is going to talk about the immunological risks of mirabacteria, and that's David Relman. David is the Thomas C. and Joan M. Merrigan Professor of Medicine and Professor of Microbiology and Immunology at Stanford University. He's a senior fellow at the Center for International Security and Cooperation at Stanford, and was an early pioneer in the study of human indigenous microbiota using modern molecular approaches. His current work focuses on stability, resilience, and evolution of the human microbiome. Among other honors, he's a fellow of the American Academy of Microbiology, a member of both the National Academy of Medicine and American Academy of Arts and Sciences. And last year, he served as senior advisor in the Office of Pandemic Preparedness and Response Policy at the US White House. He was an author of the Mirror Life Science paper and the accompanying technical report.
Thank you, James. Let me first by saying that I am the grateful beneficiary of 3 very clear, very helpful talks that precede mine and that cover a number of the principles and ideas that I would have wanted to convey. So So thank you, the three of you, and others who have contributed along the way. Deepa just explained how mirror bacteria might prove successful in the environment by acquiring nutrients, by evading predation, and by spreading into multiple habitats. And therefore, we must assume that there will be encounters between mirror bacteria and a variety of animal and plant hosts, including humans. And the means of initial contact might be multiple in the case of humans. You can imagine contact through skin and skin abrasion. You can imagine oral ingestion and even possible inhalation. And so the topic that I want to cover now is what the outcomes might be in such encounters between mirror bacteria and these various hosts. I'll touch very briefly on survival and growth, although Deepa just did a wonderful job. But I want to focus more on what might result from the efforts of our immune system to counter mirror bacteria, should they be found and then grow within the human body. I will touch briefly on potential public health countermeasures, and then speak very briefly about similar issues with respect to animals and plants, other animals and plants. And I first want to acknowledge the co-authors of the chapters that are relevant to the topics that I will address. They're shown here, as well as the other co-authors of the technical report and science paper that were published last fall, and the reviewers. So this is going to be one of slides that might be a little hard to read, and I'm just going to give you the top line here. But this slide in particular ought to be familiar to you because Deepa showed you a very similar version outlining and listing some of the achiral nutrients that might be found in relevant environments to mirror bacteria. This version of that slide now includes some numbers that have been measured experimentally from humans. And you can see by these numbers in bold that the typical concentrations of some of the more prominent and important achiral nutrients in the human body, such as glycerol or lactic acid or alanine, clearly exceed the concentrations that are thought to be minimally needed for an organism like E. coli, or perhaps others like Bacillus subtilis, organisms for which we, organisms for which we do have numbers to go by. So again, the conclusion is that there may be accessible nutrients that mirror bacteria could use in order to grow within the human body. Now, many of us would like to think that we are impervious to the occasional encounter with an environmental organism, or even, more relevantly, impervious to the possible violation of our surfaces by the billions of organisms that live within our gut, in our mouth, and skin, etc. However, such is not the case. There are now at least 50 years of experimental data demonstrating this phenomenon of leakiness. Excuse me. Humans are essentially leaky to our own commensal organisms by a number of routes that are clear from very simple experiments like taking blood cultures after you brush your teeth. One sees a burst of organisms in your bloodstream for just a few minutes, and then they subside and disappear because of your immune system that's clearing them. Similarly, even in times of relative quiet, we have organisms that are translocating across our gut at appreciable rates in otherwise healthy people. The numbers in this table here come from a study by some surgeons in Britain who undertook this heroic study in patients who were undergoing elective abdominal surgery, where they opened the abdomen and immediately took out a mesenteric lymph node to sample it for viable bacteria before proceeding with the surgery. And in this study, about 10% of all humans with no signs of fever or infection had viable organisms like E. coli in their mesenteric lymph nodes at that one moment in time. It's thought that numbers like this are typical and represent probably the adaptation that we have made and the trade-offs that we have made in order to understand our commensals and learn how to respond to them with our immune system. But notice that these organisms leaking through don't have traditional virulence factors, nor would a mirror bacterium necessarily need a traditional virulence factor because it would benefit from this low-level leakiness, but at the same time would not have to deal with an immune system or immune system that wouldn't be able to deal with it because of some of the reasons that I'm going to tell you about. So there are three aspects of our immune response that I want to touch on briefly. I will not do them justice, but for the sake of simplicity, let's call them the innate immune system, the adaptive immune system, and then some functions that are common either to both or important for host defense in general. First, with respect to the innate immune system, many of you, in fact, here at the Pasteur, some of the foundational work has been done in understanding how certain elements of our immune system recognize and then respond to a wide variety of microbes with a rapid but somewhat nonspecific response. And they do so through the role and function of what are called pattern recognition receptors. Many families, one example of which is TLR or toll-like receptors. These are receptors and pathways mediated by adapter proteins, I've shown here, MIT88 and IRAK4, that turn on a whole variety of host responses that are designed to counter whatever the microbial threat might be. So again, they're quick and nonspecific, but they depend upon these receptors recognizing molecules which are often referred to as microbial-associated molecular pattern molecules. However, in the case of mirror bacteria, these molecules, things like LPS and peptidoglycan and flagellin and CpG, DNA, etc., are all based upon and built with chiral centers. They have chiral features. Hence, TLRs and other pattern recognition receptors will not recognize them and will not respond properly. And the result of that, we understand better from experimental data. In the case of laboratory animals that are engineered to be deficient in just these one family of, or in this case on the left, one particular toll-like receptor, the outcome for mice is early death within days from infection by a wide variety of environmental organisms. This defect by itself is a profound defect for these mice. And in a similar vein, humans that are born, unfortunately, without those adapter proteins that make these toll-like receptors work well also suffer dire fates. Children that are born without the functioning of these pathways develop early in life a wide variety of infections from pneumonia to meningitis, and 40% of children die by age 8. In fact, the majority of them die in the first year or two of life. So turning to some of the effector functions, some of these might be classified within the innate immune system. So for example, the complement system is a system that, again, non-specifically binds to and mediates the killing of foreign microbes. Well, there are three different pathways: the alternative, the classical, and the lectin pathways. And because the last two of these, classical and lectin, again, depend upon the interactions of chiral molecules, such as antibody, which will be based upon the recognition of chiral antigenic determinants, these two pathways of the complement system are expected not to work. On the other hand, the alternative pathway may well work in our estimation. And there are lots of caveats here. So here's one little glint of hope. And I will say right now that there are a number of specific components of both innate and adaptive immunity that may well work to some degree. But the bottom line here is that the parts that don't, we already have good ideas of recognizing as potentially dire or highly consequential features. Another aspect of effector function are enzymes that are serving to break down bacterial cell wall, like lysozyme, or enzymes that are breaking down components of bacteria once they are ingested. And here we have, again, experimental evidence that suggests that these enzymes simply do not work when the target has chiral features, like a D peptide would. You see at the bottom, essentially no digestion. From the point of view of phagocytosis, a story almost identical to the story that Deepa told you about predation. The same kinds of steps are involved. In fact, it's believed that the phagocytic cells of our immune system evolved from free-living amoebae in the environment. And hence the same steps that Deepa outlined as all involving interactions between chiral molecules, recognition of chiral targets, and digestion, et cetera, all of these are likely to fail with respect to the action of our own macrophages and the processing of proteins, which leads to the third area, adaptive immunity. Because, as you probably know, adaptive immunity is based upon the ingestion and digestion and presentation of specific antigens to elements of our own cellular immune system so that we can develop specific, albeit delayed, responses, like antibody responses. Because the phagocytic system is not working properly, we don't expect to see mirror antigens, antigenic peptides, being processed and then bound by the relevant receptors, in this case MHC class II, and hence presented in sufficient numbers and with sufficient avidity to stimulate the target immune cell, the T cell, which is now going to orchestrate things like antibody production. We expect that T cell activation will be significantly impaired and much dampened in the face of a mirror organism that presents these mirror antigens. And again, there's experimental evidence to suggest this. In fact, the paper that John alluded to with this same figure on the right suggests that if you immunize a mouse with a D peptide or a D amino acid containing protein or molecule, you essentially see no immune response in the form of antibody production. That's the open triangles at the bottom. Again, antibody production will be impaired not just on the basis of this T cell dependent set of processes that I've been alluding to, but also as a result of dampening of the T cell independent pathways that we know about, simply because they depend upon proper inflammation signals that come through those pattern recognition receptors that I already spoke about. Again, experimental evidence, unfortunate evidence in the form of experiments undertaken by nature in humans. Babies born without just this one antigen presentation molecule, MHC class II, with a disease called Bare lymphocyte syndrome, fare terribly in early childhood. These are survival data based upon the use and the absence of the only effective treatment, which is bone marrow transplant, which, by the way, would do nothing to help us in the recognition of D peptides, for example. So focus on that blue curve, no treatment. All children with this syndrome die by age 10. There are no known survivors without a bone marrow transplant, which is not an option here. And again, remember, this is a single deficiency of a number that we have identified where we expect there to be a major defect in our confrontation with mirror organisms. The pathology that results from all of this is quite unclear to us. We are very interested in further input about this. I mean, what would the fate of human infection look like clinically? We can imagine consequences in the early or acute stages of infection and consequences from the chronic stages. In the former, there may well be a sepsis-like response simply because once our own host cells are damaged enough and break apart, we know about a damage-associated molecular pattern-based response that looks like sepsis. That likely will happen, but it will happen late, and it will happen in a very undirected and probably ineffectual way. The chronic infection aspects may well involve the inexorable accumulation of organisms that cannot be broken down properly, thereby choking cells, causing cellular dysfunction, tissue dysfunction, etc. So in summary, we believe that humans will encounter mirror organisms should they escape into the environment for all of the reasons that Deepa well described. We believe that they will be able to grow to some extent in the human body, and granted this is a really complicated set of scenarios, but we believe that they will not be adequately controlled by our immune system, and we know that some of the key defects that we more clearly expect are highly consequential defects. So at the end of the day, we end up with the sort of inescapable conclusion that it is plausible for humans to suffer as well as many other animals and plants. Are there countermeasures available? Again, Deepa referred to some of this already. There are a few available antibiotics today that are either achiral or created in racemic form, so both enantiomers are present. We expect those will be effective, but only against the random mirror organism that happens to be susceptible before either a human, either inadvertently or deliberately, makes it resistant or it evolves resistance. To make many of these other antibiotics that we might need would entail various levels of difficulty depending on whether they're made in today's world biologically or through synthetic chemical means. Bacterial vaccines, yes, we could imagine how we could construct and generate these vaccines, but it's likely that we won't have any good idea of whether they're going to be effective because we can't do challenge studies, and we'd have to guess about the nature of the immune response that would be needed in order to have any likelihood of success. However, non-pharmaceutical inventions would clearly still be effective. Could all of this be deployed on a global level? Well, look at how well we did with COVID and a good vaccine that was available in some places but clearly not available, nor were many humans willing to take it. Imagine that problem times 1,000 or 1,000,000, because the development is going to be difficult for these items, and then the testing is going to be difficult, and then the deployment and distribution is going to be difficult. So it's really a massive scaling problem. And now when we talk about the rest of the so-called higher order organisms, and I hate to call sometimes humans that higher order, but nonetheless, other animals and plants share many of the receptors that I've talked about, and hence many of the same kinds of immune responses and mechanisms. They too will likely suffer the same kinds of defects that I've talked about. And likewise in the plant world, plants, like animals, are leaky through their own particular anatomic vulnerabilities, and their immune system is also based upon the recognition of chiral molecules, and hence we imagine, although we have even less data on which to base this, that plants would not fare well. So, summary and conclusions. Upon entry, which we think is inevitable, either in a laboratory and certainly in the environment, mirror bacteria in humans could plausibly survive and grow and transgress and translocate into tissues, bloodstream, and therefore find themselves in privileged spaces in the human body where our only defense is an immune system that can work properly and sometimes the complementary activity of drugs that we have on hand. And I've already, I think, walked through many of the potential problems with that immune response, the difficulties we have with people who are immunodeficient despite the availability of active antibiotics. They die anyway. And so we have a somewhat gloomy outlook here, leading to the following recommendations that you've already heard about. First, absent compelling evidence suggesting otherwise, the authors of this paper conclude that mirror life should not be created. The authors also conclude that work dedicated to the creation of mirror life should not be funded, and that there needs to be further discussion, in part because of these uncertainties, in part because we may not have considered issues that are paramount and important for us to consider So clearly some hubris here is not warranted, and some humility is. And I will stop there. Thank you.
Great, thank you, David. So a couple of related questions have come in from people on the Zoom and in the room here today, and that's around what experiments could and should be done to understand more about this. Aren't there simple experiments that we could do to learn more, and should we be doing those before we go ahead and proceed with some of these recommendations that you made?
Yeah, really important question. In fact, we hope to have much fuller discussions of exactly that issue tomorrow. But for the sake of just jump-starting discussion or thought, first, we have to understand that there are going to be trade-offs in consideration of experiments, in part because some of the experiments we would most want to do may be not feasible, or some of the experiments we might want to do might not inform in a material way our discussion about whether we should be considering in any remote way the creation of mirror life. But clearly there could be experiments that might have some value in further instructing us. And as an example, I think you will have now understood from Deepa's presentation, John's, and what I've presented, that we don't understand as much about the fate of mirror molecules in the environment and in humans as we might need or want. And I think we could all agree that in the trade-off world, where we don't want to inspire work that facilitates and enables the creation of mirror life, we might consider the use of mirror molecules, which are already being made for all of the good reasons that you've heard about, as reagents for understanding what are the half-lives in the environment in defined circumstances. How are they processed, if at all? Are there sort of considerations that would further inform our thinking about the immune response and ecological outcomes?
Great. So related question on the immunology, and then I'll ask a couple of more general questions. There's been several points raised in the Q&A, essentially asking, is lack of recognition of a mirror bacteria sufficient to ultimately cause disease and ultimately lead to these kinds of outcomes that you're talking about? Wouldn't a mirror bacteria need virulence factors to be able to cause harm? And then a related question, couldn't humans kind of evolve to be able to deal with this?
Yeah. Important questions, because many of us thought about this for all of the good reasons that one would. From a background in bacterial pathogenesis originally, my mentor, Stan Falco, taught us about the particular lifestyle that pathogenic organisms have and the genes and the virulence factors that are needed to make a go at life in a host that's very well defended by an immune system and defended by competition with other organisms. And so here's where our considerations about relative growth and death take hold and apply in the human body. And again, the experimental evidence that we have that absent just one specific mechanism for pattern recognition, the mighty 88 deficient humans, or the MHC class 2 deficient humans, that defect alone, recognition and presentation so as to stimulate a proper immune response, that is a fatal outcome for humans.
Great. So someone on Zoom asks, have there been any examples of people strongly rejecting or otherwise opposing the kinds of recommendations that you put forward about mirror life research and in general the findings here? And if so, what was their rationale?
There are multiple hesitations and concerns about the logic that we present. And I have to say that nearly every one of these 38 co-authors, myself included, asked some of these very same questions at the start. At the start, very much doubted the overall fitness of an organism because I imagined it to be created in a very minimalistic form with many, many deficiencies, which might be very difficult to overcome or correct or restore. But I was convinced by my friends in the synthetic biology world that these are things that are already underway and things about which we know how to do. So that was the fitness, the ecological fitness of the organism was one. Another was simply the feasibility of constructing such a thing. And here again, I think John has outlined a number of the counter arguments to that, but who knows? There could be obstacles that none of us imagine that John has alluded to that prove to be far more difficult than one might think, and that could take much longer than we think. Could also go the other way. So fitness, feasibility, and then the outcome that I've talked about. Again, I think the most convincing thing for me is the evidence from nature, those deficiencies in humans where despite their imperfections as models for what we're talking about, they're close enough to give grave concern.
And do you think that bringing this work forward, publishing this paper, talking about these risks, including in settings like this, could constitute an information hazard? So is bringing this risk forward potentially actually causing risk in and of itself?
Yes, I think there is certainly the possibility of some hazard here. A number of people, again, raised that to us in the early days of considering what to do with this assessment. What convinced me that we were better off speaking out now were two things. The first, that there already has been, as you've heard from Filippa and others, a long-standing interest in doing exactly this. And now, increasingly in recent times, raised by scientists who had been pretty determined to pursue it. A grant from the NSF asking for the early steps towards the creation of mirror life. Lots of other scientists that I've spoken to, and many of you here perhaps are included in or know of, who thought this is a wonderful dream for humans to pursue because of what it would say to us about humans with this undying curiosity and this incredible ability to overcome technical obstacles. But it's all of these people out there already talking about it. And then quickly, the other is that we have time now. It is, as you've heard, near impossible for any one person to undertake this now without at least being noticed for their interest. And this is the time when we should be taking advantage of this lag that we have, that many sociologists and people in risk have always dreamt about having in order to spawn the kind of discussions we're having and societal input before we have the real means of executing.
Great, thank you, David. So for the last 10 minutes of this discussion today, Professor Yasmin Belkay, the president of the Institut Pasteur, who has been a key part of this work and setting up this conference, a co-author on the Mirror Life paper that came out last year that we've heard about today, unfortunately at the last minute is unable to attend. So in her place, David Relman is going to provide some concluding remarks, bringing this all together from the perspective of a co-author of this work, followed by David Bikard, who will give the perspective from the Institut Pasteur. So back over to you, David.
Thanks, James. And needless to say, we would love Yasmin to be here. We hope that she can listen to what's transpired, and we certainly look forward to her input. Well, I offer these comments with a great deal of humility. I want to make clear that there are some huge uncertainties about the assessment that we have proposed. I think Deepa said it well. We're not asserting that these conclusions about risk are a known fact. We're simply asserting that they're plausible. And that, coupled with the magnitude, the potential magnitude of the threat, I think, compels us to address this now, and in the absence of information otherwise, to conclude for the time being that we should not proceed towards the creation of mirror life. Mirror peptides aside, different story. We've heard this afternoon that although they are not an imminent threat, mirror bacteria could be a threat of extremely serious consequence stemming from unique immunological and ecological risks. I've spent a great deal of my career on the side thinking about biosecurity and about plausible and imagined risks. And I have to say that until now, I think many claims of existentiality or calamity have often been overblown. And I have been happy to call that out. But this is now, I think, the first case where I have to acknowledge that there is a plausible risk of an existential sort. And I'd love to be shown wrong or have a good argument made that that's not the case. In the time since the paper was published this past December, there's been a great deal of engagement with this work. And to this point, broad agreement that the risks clearly outweigh the benefits. The response of the research community has been remarkably encouraging. You've already heard that at the recent 50th year anniversary of the Asilomar Conference, many of whose participants are here in this room, nearly 100 of the participants from over 20 countries signed a statement agreeing that mirror life should not be created unless future research convincingly demonstrates that it poses no severe risks. And in the wake of that, and in the midst of that, I just want to offer my appreciation to those specific scientists who previously had the goal of creating mirror life, who now have joined this effort to think through the risks and ultimately argue that mirror life should not be created. They have essentially sacrificed their own research and personal goals. They certainly could be considered role models for this. And I think that, at least so far, many in the scientific and broader research and policy communities have recognized this. We have a rare opportunity now in which the risks of a technology have been recognized far in advance of its development. And that means at a time when we have a real chance to stop this work, at least for the time being, and to consider it with great care. But that window, this window of opportunity, is not indefinite. One of the things that worries me is that we will not take advantage of this opportunity. Either we'll say that we don't need to address this risk now because there are many more current, pressing, urgent interests, priorities. Or we may wait too long because people argue that we need to be totally certain before we move ahead with something like banning scientific work. In either case, we will have lost what I think is a rare opportunity to act in an anticipatory fashion and prevent an unprecedented risk from materializing. And we also have an opportunity to demonstrate the willingness of scientists and others to come together ahead of time, recognize and talk about a potential large risk, and accept responsibility for it, meaning that with this power that scientists have will come responsibilities, and the acknowledgement of these responsibilities means to accept limits on that power. And as a spin-off, I can see that accepting and embracing limits will mean that scientists might be taking an important step towards rebuilding public trust in science and scientists. So I think this is one of the few opportunities that I can think of where we have this kind of setting in which to hold a meaningful discussion and talk about what this means and what to do about it. And that's, of course, why we're here. We're here to work together to recommend further actions. And I desperately hope we don't lose this opportunity or fail to take it seriously, so that we can do this in a timely manner and basically demonstrate the willingness of scientists to do the right thing. So thank you all for being a part of this discussion.
All right, so I realized this morning I didn't introduce myself. So I'm David Bikard. I'm the head of the synthetic biology lab here at the Pasteur Institute. And so it was an amazing pleasure and to host this historical event here. I'd like to thank all the speakers for their exceptional presentation and insights. And of course, the organizing committee from the Microbiology Dialogues Funds for putting all this together. I'd like to thank you all for being with us today, to all the attendees online. We had over 300 registered participants online, and it was great to see the very active discussions. I'd like to conclude this also with a more hopeful view of the scientific enterprise and all the progress. That is also the reason why we're having this discussion now. Just to give a figure, I think it's around 50% of the new drugs that are brought to market that are biologics, which are the direct product of recombinant DNA, genetic engineering, and I think it's critical that we pursue this scientific enterprise to bring the benefits to humans, but also do it in a responsible way. We're incapable of building moral life now. And the point of having this discussion now is to make sure that 20 years from now, 30 years from now, we're still incapable of doing it, and we can make sure that everyone is safe. Today's meeting is a part of a much larger discussion that's taking place around the world, around moral life. There will be follow-up meetings in Manchester in September this year, and another one in Singapore in the spring of 26. So I know I'm standing in the way of a glass of wine at the reception, so I will end here. I look forward to discussing with you at the reception, and please join me again in thanking all the speakers for their presentation, and thank you for your participation.